Yo, what is up, iGEM gang? So today we're going to teach you guys how to use Snapgene, which is going to be a very important program that you guys have to use in order to construct your plasmid. So in order to make a plasmid, just as a quick review from class, you need a gene of interest. And in that gene of interest is basically creating a protein that does a function, and that function could be anything. For example, if you want to make a bacteria that luminesces, right, you want a gene that produces some sort of a luminescence. So that is where your gene of interest comes in. In order to have a gene of interest, you need a promoter DNA sequence. This DNA sequence obviously kickstarts the production of that protein, the transcription and the translation. You need your ribosome binding site, which is where the ribosome binds and then produces that protein. The gene of interest, so obviously this is going to be the DNA sequence that produces that protein, and then the terminator, which is basically a DNA sequence which ends transcription. So all of these DNA sequences in this specific order is very important in order to have a functional gene that produces a successful protein. Now, in order to carry this gene of interest into your bacteria, you need a backbone. So a backbone is a long circular DNA sequence which contains some sort of antibiotic resistance. So it serves two functions. It is to carry your gene of interest, but also to have some sort of antibiotic resistance. So for example, my backbone can have ampicillin resistance. So what's the point of having antibiotic resistance? So the point of it is basically when you're plating your bacteria, right, you want to kill off bacteria that you don't want. So you want to kill off bacteria that is going to harm the production of your bacteria. So basically when you're plating, right, so you could have an ampicillin plate or a kanamycin plate or any sort of antibiotic, depending on your project, you want to kill off the bacteria that don't have any sort of ampicillin resistance. You want your bacteria to be the only surviving bacteria. Thus, you want to have bacteria with some sort of antibiotic resistance. So in this case, let's have a backbone with ampicillin resistance because we want this bacteria to be the only surviving bacteria when we plate our bacteria. When we combine our gene of interest in our backbone, remember we scissor our backbone with restriction enzymes, which we've learned in class, and we ligate our plasmid and we ligate our gene of interest and our backbone together to form our plasmid. And this plasmid then enters the bacteria to then conduct its function. So how do we find our backbone? How do we find our promoter, our RBS, our gene of interest and terminator? So to find your DNA sequence for your promoter, your RBS, your gene of interest and terminator, you can use websites such as the iGEM Registry, which we're going to show you how to use in a little bit, and NCBI, so you can blast and figure out what gene you want to use. So we're going to show you guys how to use this website. For your backbone, you're going to use a website called Addgene. So Addgene is a website where you can find many different plasmids and backbones. So we're going to show you how to navigate through Addgene. So once you find your DNA sequences from the iGEM Registry, NCBI, and Addgene, you're going to take all of these DNA sequences and then you're going to put it together in a plasmid using Snapgene. And Snapgene is a program that you guys are going to have to download, and once you download it, we're going to teach you guys how to use it. What's up, iGEM gang? So we're back, and the first thing we're going to do is we're going to look for our backbone. So as we've mentioned, the backbone is used to carry our gene of interest, and it also carries our antibiotic resistance. So the website we're going to use to figure out what backbone I want to use or you would like to use for your project is Addgene. So Addgene is a pretty simple website to use, and it's a very great way to find your backbone. So in order to find your backbone, what you want to do is obviously go on addgene.org.
And once you've entered the website, you want to go on Browse Catalog. On Browse Catalog, you're going to see the type of DNA sequence you want. So you have your plasmid, a viral service, DNA service, plasmid kits, and all that. So what you want to do is you want to click on plasmids. Once you've entered plasmids, then you want to scroll down and you want to go to where it says Browse by Expression System. So if your project is using bacteria, or your project is using yeast or mammalian cells, you want to pick which plasmid type you want to use or backbone type you want to use. So let's say, for example, I'm working with E. coli. So E. coli is obviously a type of bacteria. So I want to go on bacteria because that is the type of expression I want to use. So now you're going to find a bunch of plasmids that are compatible with E. coli or any kind of bacteria. So you can basically just kind of scroll down and just sort of navigate through this and sort of take your time to kind of find which backbone works for you, and to kind of like find out what genes are in each of these backbones. So you can also kind of narrow down your search results through this. So I just want to, so as I mentioned, I just want bacterial expression. For vector type, I want to just use, you could use synthetic biology. That would kind of narrow down our search results a little bit more because we're just using this for synthetic bio. For species, what type of bacteria do you want to use? So if you're using E. coli, obviously you want to narrow down your search results to E. coli. So now it went from like thousands of search results to 200 search results. Now there are even more filters. If you want your bacterial resistance or antibiotic resistance to be ampicillin or kanamycin, you can narrow down your search results even further. So in this case, I'm going to narrow down my search results to ampicillin because I think ampicillin resistance is just pretty common and it's very standard in a lot of synthetic biology projects. But again, this is going to be very dependent on your project. This is just kind of an example. So now that I've narrowed down my search results by using the filter, I can find whatever plasmid or backbone I want. So I'm gonna scroll a little bit and basically just kind of like go through each of them and just sort of take your time and kind of like understand what each of these and understand which backbone is the right one for you. So I'm not going to do that in this video, but what I'm going to do is I'm going to just pick a random backbone. So I'm going to pick this PET21 backbone. Honestly, plasmids have really weird names. Yeah, it just is what it is. So here it is. So down here, you're going to find a lot of information about the backbone. So we know that it has ampicillin resistance. So if we grow this bacteria in a plate, we only want the bacteria with ampicillin resistance to survive, and we want to kill any bacteria we don't want. This works in E. coli that is DH10B species. So every E. coli species is kind of different. So be very careful on what species of E. coli you're using. So if you're using DH5 alpha E. coli, make sure to pick DH5 alpha. Also, it produces a high copy number. So it's pretty good if you want to make a lot of your plasmids and you want to make a high copy number of your plasmids. So we know that the species is E. coli. So let's say that this is the backbone I want. I choose this backbone. Again, this is going to take some careful consideration on your part, but for this lesson, let's just say, for example, that this is my ideal backbone and this is the species of E. coli I want to work with. So what we want to do is we want to enter this backbone right here into Snapgene. So what we're going to do is we're going to click enlarge. And so this is the backbone right here. And down here, it says Snapgene. So you want to click on Snapgene. And this right here is going to download a DNA file that's going to contain the sequence for your backbone. So I'm going to open it up. All right. So once your sequence has downloaded, your backbone sequence should be installed and it should open up on Snapgene. So here is my DNA sequence for my backbone. So I downloaded my PET21A backbone, assuming that this is the backbone I want to use, obviously. And as you can see, there are different genes in here. So all of these arrows right here are the different genes that are within this. So it contains the ampicillin resistance gene. It contains a LAC1 gene. Some of these stuff you're going to have to kind of look into to make sure that it's okay and it doesn't harm or interfere with your with whatever you're trying to do. But these arrows are just meant to represent the very important genes that are within this backbone. And right here, all of these letters, you may be wondering what these are. Those right here are your restriction enzymes. So if you remember from class, your restriction enzymes are basically the cutting points. So these are points where your backbone is going to be scissored and cut. So just think of these as like scissor points where you're going to cut your backbone. And when you cut your backbone, remember, you're going to tape in your gene of interest. So if you want to insert a gene, for example, on making your bacteria glow blue, for example, right? You're going to take two restriction enzymes. So I could take this right here and this right here. So I could take PVUI and BSA, and I can cut this region right here. So I can take this region and I can cut this region. And then in place of that region, I can put in whatever gene I want. So yeah, that is how to navigate Addgene and to then get your backbone. I'll see you in the next part.
Okay, so today's video is just going to be a very brief overview to the registry of standard biological parts. And this itself is a really good tool for finding different promoters, terminators, RBS that you'd like to incorporate into your construct. But also keep in mind that this is by no means a limitation if you can't find a specific coding sequence that you would like to incorporate. A big part of iGEM is also creating new parts and improving upon old parts that you have. But that's kind of getting ahead of ourselves. So let's start with just navigating through a parts page that we see right here. This is the page for GFP, and GFP is a very common reporter gene used in a lot of different synthetic biology projects. So starting at the top, we have the name for it, and an iGEM part is always going to start with BBA and then some combination of letters and numbers. And that's just how they're characterized. And then you're going to have your title. So for this, we have the green fluorescent protein. And this main page itself contains a lot of information about contributions. So how previous iGEM teams have been able to use this specific part. So an important thing to keep in mind if you're using this part in your construct itself is if it's compatible with the chassis that you're using. To find this information, you can click on the information tab right here and scroll down to categories. And here you see chassis and then D. subtilis and E. coli. So you know that it's compatible to be used in both of those. If you don't see that information there, you could also potentially find it in design notes or within the main page. If you scroll through contributions, you might be able to see information on how other teams have used, which types of chassis other teams have used this in. Okay, now that you have your information and you know that this part's going to work in the construct, how do you get the sequence? You can also click on design again and click on get part sequence. Great. Now you just have the sequence. You can copy and paste it directly into Snapgene. Very convenient. And that's it. And if you see this ruler right here, for something like GFP, because that's the only sequence we have in this specific part, it's not really going to come in handy. But if you have a composite part, which is basically these individual parts, so pretend that one of these things is GFP and you have some other part that's there. So this composite part is basically just made up of smaller basic parts that are linked together for some type of relevance. And in this case right here, you have basically an insert that's ready to be put directly into your construct, as you also have just the promoter, terminator, and RBS, as well as your coding sequences. So that brings us to navigating for those specific parts, so promoters. So we can go to promoters itself and click on catalog, and you can find that you can either navigate based on the function you want, say if you want it to be constitutive and just on by default, or any other function that you'd like. Or you could also navigate based on the chassis that you'd like to have it in. So we have things for E. coli, B. subtilis, and then for eukaryotic, yeast is most commonly used within iGEM. So you have information for that. And then that's basically how it's going to be for promoters and terminators. And then for RBS, you also have it split based on chassis. So for E. coli specifically, the Anderson RBS family is used a lot. So you can really just navigate through the library and find out and just use any one of them if you're using E. coli, or if your RBS is actually compatible with your construct. And going to coding sequences, either if you have a coding sequence that you would like and you found it from an iGEM project itself, if you go to parts pages on the parts tab on their wiki, you might be able to find a link to a parts page. Or if you find it from some paper, you might be able to find the sequence from there, and there's other things that you could do to be able to use that into your sequence. Or if you just specifically want a general function, say a reporter gene, you can find it in the protein coding sequence tab itself. And you have things separated into enzymes, protein coding sequences, all different types of things. So for reporters itself, you have a lot of different options. And I think my main point in this is that it's kind of, I can't encompass everything about this about iGEM parts into one specific video. So I would highly recommend for you guys to look at the parts wiki independently and to really just get yourselves comfortable with it, as this is something that you're going to use a lot throughout the summer and as you're developing your project. So just getting comfortable with it, understanding maybe what you'd want to use, if that's something, it's quite important. That's it.
All right, so as we've learned in the last part, we were able to navigate the iGEM registry and NCBI, and now we're able to pick out our parts. So let's say I want to make a gene that makes a spider web, or I want to make spider silk. So as we've learned in class, in order to do it, right, the gene for spider silk itself is not enough. You need more in order for this to work. So obviously you need your promoter DNA, your ribosome binding site, obviously your spider silk gene, and then your terminator. So the promoter, ribosome binding site, terminator, I was able to find the DNA sequences on the iGEM registry. And then for the spider silk gene, I was able to find that on NCBI by conducting a blast. So let's open up the promoter sequence, for example. So once this loads up, so right here is the page for your promoter. So this promoter is used in E. coli, and it's compatible with E. coli. So what you want to do is you want to go on get sequence right here. And right here, you can see your sequence. So obviously DNA is made of guanine, adenine, tyrosine, and cytosine. So you're going to see these GG, CCs, A's, and all those sequences. So here is my DNA sequence for my promoters. I'm going to take this and I'm going to copy and paste the sequence. And I'm going to copy and paste it onto a Google spreadsheet. So I'm going to take my promoter and I'm going to put this on a Google spreadsheet. So right here where it says promoter, I'm going to take that and copy and paste it. So, all right. So as you can see, I got my promoter, my RBS, my gene of interest, and my terminator sequences all copy and pasted onto a Google spreadsheet, just for convenience and organization. So once I have all these parts, I'm going to put them together on Snapgene. So what you're going to do is you're going to take your sequences, you're going to copy and paste them, and you're going to take them, copy and paste them, and put them onto Snapgene. So what you're going to do is you're going to open up Snapgene. So I'm just going to close this window. Sorry. And here we are. So we're going to open up Snapgene. And what we're going to do is we're going to click on new DNA or RNA file. So here we are. We open up this file right here. So obviously we're working with DNA. So we're going to click DNA. And what we're going to do is we're going to copy and paste our promoter, RBS, gene of interest, and terminator all into this. So we're going to create a sequence. So basically it's like Lego bricks. So we're going to put it all together. So I'm going to put my promoter in first. Then I'm going to take my RBS and paste that in here as well. Then I'm going to take my gene of interest and just keep copy and pasting it until you have all your sequences that are together. And also, last but not least, but your terminator as well. So now we have our entire DNA sequence for a spider silk gene. So now that we have all of this, we can title this gene. So I'm going to title this spider silk gene. Done. So we created our spider silk gene. Okay. And then it's going to open up a new window that's going to show our DNA sequence altogether. So here we are. We're going to click yes, add this one feature. So here we are. So this right here is going to be our DNA sequence. So right here, you could see our full sequence right here. You could see all the enzymes that are on here, etc. So yeah, here is our spider silk DNA. And basically this entire DNA sequence right here, this is the map. This entire DNA sequence, what we're going to do is we're going to take this and we're going to put it onto our backbone. So here it is. All right, so now that we picked our backbone, now that we constructed our DNA insert for spider silk, now we're going to take our insert, which contains our promoter, RBS, etc., and now we're going to plop it onto our backbone. So what you're going to do is you're going to open up your sequence for your backbone, which we did already on Addgene. So you're going to open this up, and what you're going to do is go on top and click here where it says actions. Once you click on actions, go down and you're going to see two options. You're going to see Gibson assembly and NEB builder hi-fi DNA assembly. So for iGEM, though Gibson assembly is technically the standard, for iGEM, you're going to be doing hi-fi to put your backbone and your insert together. So you want to go on NEB builder hi-fi and you're going to insert fragment. You can insert as many fragments as you want or insert as many DNA sequences as you want. But since we're working with one DNA, we're just going to click here, insert fragment. And it's going to lead us to this page. So right here is our backbone. And what we want to do is we want to cut open, we want to cut our backbone. So to cut our backbone, what we want to do is we want to pick two restriction enzymes. So these two restriction enzymes, we're going to cut it. And once we cut that region, we're going to then plop in our DNA insert. So what I'm going to do is click here where it says linearize with restriction enzymes. All right, so now I'm going to pick two restriction enzymes. So I want to pick, so let's say I want to insert my DNA somewhere around this region. I think this looks like a pretty good place to cut and insert my DNA. So what I'm going to do, I'm just going to click linearize right there again. And I'm going to pick two restriction enzymes. So let's say I want to pick this and this. So I picked PSHAI and BPU. So I'm going to cut within this region right here. Okay. So this highlighted region over here is where I'm going to cut, and that's where I'm going to plop in my DNA sequence. You want to then go up where it says fragment. So click on fragment. And for source of fragment, I already picked mine. So click on whatever fragment you want to enter. So obviously, I'm going to insert my spider silk gene, which I'm going to plop into my backbone. So here it is. So there's my spider silk gene. So once you've done that, make sure use as a template for PCR is selected, and make sure that the second option right here creates overhangs, 3 in G, A or G overhangs is selected. So once you pick that, you can then move on to products. So what you want to do is you want to pick your bacteria species. So let's say, for example, you're going to use DH5 alpha, just pick that. This may depend on your project, but for this example, we're just going to pick DH5 alpha bacteria or E. coli. Next, what you want to do is you want to make sure you go on your fragment and you press Control A, or I'm not sure what the command is on Mac, but you want to make sure that your entire sequence is highlighted, and you'll go on your product. So we picked our bacteria species, and now we want to click on choose overlapping PCR primers. So now we're going to develop our primers, which is going to be the sticky ends, which will stick onto our backbone. So what we did was we first cut our backbone. Now we're creating the tape, which will stick our insert onto the backbone, creating a final plasmid. So once I've done that, I'm going to click choose primers. So these you can adjust depending on your project, but just for the sake of this video, I'm going to kind of keep it simple, and we're going to just choose our primers. Once you're done, you're going to have your assembled DNA right here. So now this right here is your assembled plasmid. So as you can see, this red region right here is our gene for spider silk. So as a result, we took our backbone, we cut it open, and as a result, we inserted our new DNA sequence. And that right there is the tutorial for Snapgene. So if you got any questions, we're going to be going over this more in detail in class. So yeah, have a good day, guys.